Hey everyone, Dr. S here, bringing you today's lecture, which is amino acids. Today's learning goals will be first, to apply your knowledge of acid-base equilibria towards predicting an amino acid's protonation state. If you're still a little shaky on acids and bases, that's okay. It's a really challenging topic. I'd recommend maybe pausing here and going back to the previous lecture and a half, and rehashing. Once we get a feel for that, the next thing we will be doing in today's lecture is practicing how to predict the properties of an amino acid going off sidechain structure, its protonation state and charge, composition, so forth. And once we're able to do that, we can begin to have a conversation on why amino acids are so uniquely suited as the building blocks to proteins. Moving forward, we will be doing some simple Henderson-Hasselbalch type calculations. So if you're not comfortable guesstimating logs in your head, why don't you take a moment, pause the video, and dig out a calculator or the one you use on your phone to help you in the upcoming slides. Today's molecule of the moment will be 2-amino-ethanoic acid. You may know it by its more common name, glycine. It is the simplest amino acid. And to be an amino acid, all you need is an amine group and a carboxyl group on the same molecule. Both of these functional groups have titratable protons on them, which means they will have pKa's, and they are respectively pKa of 2 and a pKa of 9 for the carboxyl and amine group. Now, juxtaposed to glycine, I'm going to also show you two similar molecules. Acetic acid, which I think all of you probably loathe at this point, and ethylamine. They are, think of them as like fragments of glycine. They both exist off of a 2-carbon backbone, except acetic acid just bears the carboxyl part, and ethylamine just has the amine functional group on it. And their pKa's are different. For acetic acid, it's 4.76, but let's just say 4.7 to make it simpler. And the pKa of ethylamine is 10.8. Remember how I ended last lecture? How pKa's are considered to be constants, but they can vary dramatically molecule to molecule, molecule to context. Well, this is a great example of that. Glycine has common functional groups with acetic acid and ethylamine, yet all of their pKa's are very different. My questions to you are, which of these carboxyls are most acidic? Which of these amino groups are most basic? And try to explain in a sentence or two why 2-amino-ethanoic acid would have such different pKa's. Pause the video, try to figure it out. I encourage you to use the internet because this is going to be tricky, and we will go over this first thing in class. And because this question is quite tricky, I'm going to give you an additional hint. Consider the molecular structure of glycine after it has undergone that first deprotonation. And in that, there may be a clue to answering the third part of this molecule of the moment. Going back to the basics, I'll reiterate that technically any compound that has an amino group and a carboxyl group on the same molecule can be considered an amino acid. And some common properties generally shared by the amino acids are that the pKa's of the carboxyl and amine respectively are 2 and 9. This is important because it means at neutral or near neutral pH, such as physiological pH, they will both be charged. The amine group will be protonated and positively charged, and the carboxyl group deprotonated and negatively charged. Now, bridging both of these functional groups will be a central tetrahedral carbon center, and we sometimes call this the alpha carbon, which is a term that you should become familiar with. Tetrahedral alpha carbon center. And this arrangement of having an amino and carboxyl group attached to the alpha carbon allows for amino acids to polymerize into polyamides, aka polypeptides. And they will do so in the following manner. The carboxyl group on one amino acid will react with the amine of another amino acid, like so, and they can combine to form enamide bonds, and in doing so, we will release one equivalent of water. Hence, this is called a condensation reaction. How many of these amino acids can we stitch together in this manner? Well, the sky's the limit according to biology. Titin is the largest protein currently known to science, and it dials in at 36,000 amino acids all lined up in a row. The molecular weight of that protein is an astonishing 3 million Daltons. So these amino acid chains can grow quite big. Also attached to the alpha carbon will be a simple hydrogen atom, which we can more or less just disregard. What is more important is the yellow R group shown over here. And the R group is what confers functionality to the amino acid or the polymers of amino acid. And the R group can be something as simple as a hydrogen, as shown in the case with glycine, or they can be quite intricate and contain one functional group or several functional groups. You may vaguely recall the 20 proteinogenic amino acids that were introduced to you in general biology. These are the ones comprising proteins and are translated from our genetic code. We'll spend most of our time discussing these amino acids. Some examples include lysine and glutamic acid. However, I also want to briefly introduce to you some non-proteinogenic amino acids, of which there are hundreds. Some amino acids are critical for protein function, yet they aren't genetically encoded and need to be engineered in after the fact, post-translationally. An example of that is 5-hydroxylysine. This residue is abundant in collagen, and collagen is 70% of our skin by dry weight. A vitamin C dependent enzyme is responsible for inserting this hydroxyl group into the 5th position, and that's what helps keep collagen fibers hydrogen bonded together. Without vitamin C, we can't put this hydrogen bond donor and acceptor into this spot of the side chain. And as a result, the cohesion between collagen fibers will suffer. And that would cause the collagen fibers to unravel, and that's why your skin falls off when you have scurvy. It all comes down to amino acids. There's also a GABA-aminobutyric acid, GABA for short. This is an example of a class of amino acids that aren't used in protein synthesis at all. Instead, it's an inhibitory neurotransmitter. And what's interesting is that glutamic acid is also a compound that moonlights as a neurotransmitter, but that does have a role within protein synthesis. Some amino acids are not friendly, they're toxins. Hypoglycin A is a great example of that. It is found in the unripe ackee fruit, which you can find in certain places in the Caribbean. It completely inhibits fatty acid metabolism and has an 80% kill rate if not treated. We'll learn more about that towards the end of this course, actually. And lastly, some amino acids are completely synthetic. Probargyl glycine has a very unusual alkyne in its R group, which is a functional group that's not really found in biology. And this is valuable to scientists because we have ways of hacking in this amino acid into specific proteins. And this would then allow scientists and researchers to make modifications specific to that protein by reacting it in a way with reagents that are specific to binding alkynes. So I hope that I've convinced you that even as single entities, amino acids can be highly functional. On to the main event, the proteinogenic amino acids. My goal is not to describe them individually. You will be memorizing their structures, names, pKa's, and single letter codes outside of class. Instead, right now, we'll be highlighting the general trends and properties. As I said before, there are 20 proteinogenic amino acids, 10 of which, half of them, are classified as nonpolar. Besides tyrosine and methionine, which have phenol and thioether functional groups, these nonpolar amino acids are not reactive. None of them are very water soluble once they're arrested in a polypeptide backbone. So why do you think nature evolved so many different nonpolar side chain geometries? I mean, take a look at some of these R groups. Isoleucine and leucine have very similar hydrophobic chains. I mean, they're isomers, is right in their name. Not a huge difference between alanine, whose R group is a methyl, and glycine, which is the same thing without the methyl. So what gives? Think about it. Well, the reason, and it's a good reason, is that having all of these different nonpolar geometries, that's what's largely responsible for dictating a protein's shape. This is sort of like a library of puzzle pieces where we can maximize side chain packing while minimizing steric clashes between them. I actually studied a protein whose folds would depend on a small alanine interlacing with one valine, like that. And if these two amino acids could not line up properly, like let's say if we've had another valine over here, look at that, now we have a steric clash, and the protein wouldn't fold properly at all. The protein actually would assemble into nanotubes, and by substituting that key alanine with another valine, it completely wrecked the protein. So the nonpolars have this reputation of being boring and forgettable, but in my opinion, they're kind of the most important ones. The next class of amino acids have polar side chains, and we can further subdivide these into the nucleophiles and the non-nucleophiles. Within the nucleophilic camp, cysteine may be a familiar residue. Perhaps you recall its ability to oxidize with other cysteines to form these disulfide bridges, and these are very common structural motifs in all sorts of proteins. In addition to that, the thiol in the R group of cysteine is quite nucleophilic, which means there are several enzymes that will employ cysteine residues to do chemical reactions. In addition to that, we have two other important nucleophilic residues, namely serine and threonine. They both have hydroxyl groups in their R chains, and they will also serve as reaction nucleophiles in many cases. We will be spending about 1 to 2 lectures discussing how an enzyme called serine protease functions later on. So serine is very important in this course. Moving on to the non-nucleophiles, we have glutamine and asparagine. All right. Sometimes they're called the amido amino acids because they contain amides in their side chains. They are considerably less reactive than the nucleophilic residues, but they're still useful. Their primary role is to help stabilize local protein folds and arrangements through hydrogen bonding. These are pretty good hydrogen bonders. They can also be persuaded to react with sugars to form glycosylation sites, and they can even condense with each other to form crosslinks called isopeptide bonds, but we won't be discussing those as much. The last classes are the basic and acidic amino acids, and more so than the polar amino acids, these are crucial in helping proteins stay solubilized, all right? The hydrophobic amino acids you're going to find in the interior of a protein, kind of like a micelle, and the polar and charged amino acids, such as these, are going to be preferentially decorated on the outside. So let's talk about our basic amino acids. These all contain nitrogen and there are groups. We've got lysine, arginine, and histidine. Histidine is a little weird. Let's put a pin in that and focus on lysine, for example. Lysine has an amino side chain that's on this pretty lengthy, flexible hydrocarbon tail. It's also pretty famously nucleophilic. It's a really good nucleophile. Let's compare this to arginine. Arginine is a very bad nucleophile, but it's considerably more basic than lysine. Take a moment and try to figure out about how much stronger is arginine as a base compared to lysine, going off their side chain pKa's. The pKa of this guanylyl group is 12.5 compared to the 10.5 of the primary amine and lysine, 2 orders of magnitude bigger. That means arginine is 100 times more basic than lysine. So they have different strengths. Arginine is going to be really good as serving as a non-nucleophilic base, and lysine can function as a base, it can function as a nucleophile, it can be pretty flexible. Now at the bottom, I have glutamic acid and aspartic acid. And I'm going to blow your minds and say that these are the acidic amino acids. Big surprise. They have carboxyl groups in their side chains, as well as carboxyl groups on their C termini. Now in the middle, we have histidine. Most textbooks are quick to call this a basic amino acid, such as lysine and arginine. And I get it. It has an imidazole side chain that has a titratable nitrogen atom with a pKa that is 6. All right. In later chapters, it will become apparent that histidine can act as both a base and an acid. And I think that pKa foreshadows that. It's closer to neutrality than any other sidechain pKa we've seen so far. And this means the equilibrium between protonated and deprotonated side chains will be the least skewed. So histidine is going to have this ability to sort of moonlight in both camps, which I think makes it really cool. Let me explain that a little bit further in the next slide. To get a better feel for that, let's consider the protonation equilibria of histidine alongside something more obviously basic like lysine. Over here, I've generalized the Henderson-Hasselbalch equation, and I made a slight adjustment. I just wrote deprotonated and protonated in this ratio term. It's the same thing as A minus over HA. Or in the case of a basic residue, B over BH plus. Same thing, really. Now, what I want us to do is compute the percentage of deprotonated side chains in a histidine solution compared to a lysine solution. And both will be buffered at pH 7. So let's take a minute to pencil down our answers here. I will model out the first calculation, and I encourage you to pause it when I'm done and try to preempt me on the second one, and then you can compare our answers. So it's a pretty simple setup in my opinion. The pH is 7 if we're working off this first problem. The pKa of histidine is 6. I expect you to have that memorized sooner rather than later, but since you just met histidine, I put it over here in the question, plus the log of deprotonated over protonated. And I didn't tell you what the actual concentration of the solution is, and frankly, it's not relevant. So we can whip out that trick that I showed you last time where we just solve for the ratio, what I call, big X. Simplest math ever, 7 minus 6 is obviously 1. Log of x. Log of x equals 1 must mean that x equals 10. Not just 10, it means 10 equivalents of deprotonated histidine, neutral histidine, will exist in solution alongside 1 equivalent of protonated, positively charged histidine that has undergone the acid-base reaction, 10-to-1 ratio. Now, we're going to try the next question, finding that ratio for lysine instead. pH is still 7. The pKa is quite different. It is much higher, 10.5, which makes sense because a higher pKa should mean that we're dealing with the stronger base, aka weaker acid, plus the log of x. This works out to negative 3.5 equals the log of x. Put everything to the power of 10, and that should equal about x equals 0.0003 lysines with a neutral side chain for every 1 lysine that has a protonated, positively charged lysine that had undergone the acid-base reaction. These ratios are considerably different. In theory, both histidine and lysine are so-called basic amino acids, but compare what they look like at equilibrium at pH 7. In the case of histidine, about 90-ish% of all side chains will be deprotonated, and they would not have donated that proton on their imidazole functional group, and only 10% would have a positive charge on it. Compare that to lysine, it's going to be the inverse. The vast majority of lysine side chains will be positively charged. Only about, in this case, it looks like it's 1 to 10,000. 1 out of 10,000 lysines are going to be neutral, a pretty small minority. So I hope after doing a little bit of math, I think we can now appreciate how borderline of a base histidine is when you compare it to lysine. And it's a really powerful skill to be able to look at an amino acid and instinctively know if it will be charged or not at a given pH. So I highly recommend that you train this muscle. If you prefer math over visuals, it might be easier to see an amino acid's relationship with pH in a titration plot. And I've shown you this experiment before, but what we are going to do again is add glycine into an acidic solution, dissolve it, and to that solution, we will slowly add in base sodium hydroxide solution, drop by drop. And as we do that and progress along the x-axis, we will track how the pH changes in the y-axis using a pH probe, an electrode. And so in this plot, we always begin at low pHs. This is the start. And notice the protonation state first of glycine at extremely low pH. The carboxyl group is protonated, the amino group is protonated. It's maximally protonated. And be aware of this subtlety. When you protonate a carboxyl, it does not have a charge. You protonate an amine, and it acquires a positive charge. So the net charge in glycine at this configuration is +1, because we're driving protons onto the entities in solution. We're going to start adding base through our titration experiment, and in the beginning, the pH will not be changing very drastically. Why is that? Well, glycine is buffering the solution. Yes, glycine can be a biochemistry buffer. It is the pKa of the carboxyl group that is serving as the buffer over here. And if we follow this across, we can sort of confirm that because the pKa corresponds to a pH value of around 2. And we know that to be the case for the carboxyl groups that are on the side, the C termini of amino acids. Now at this point, what fraction of the carboxyl group would be deprotonated? Remember, the pKa is the pH where half of the species have undergone deprotonation. So right over here, we are at 50% COOH and 50% COO minus. Now past this point, the carboxyl's buffering capacity begins to suffer and gets used up. And then we approach the first endpoint, if you will, of this titration. And the point where the pH of the solution is least stable, where the slope of the pH curve is steepest, is right over here. And this corresponds to the complete deprotonation of the carboxyl group. And at this stage, our glycine has a net neutral charge because the positive charge on the amine is counterbalanced by the negative charge on the carboxyl group. And when we reach this point where the net charge of our amino acid or protein is neutral, we call that the isoelectric point. And this is an important concept, particularly for proteins, because once the net charge of a protein becomes neutral, the solubility of that protein takes a pretty big hit. And that is sometimes a trick that we use in the lab to shock it out of the solution, and we can selectively precipitate proteins this way, which is a handy way of isolating them and purifying them. But for now, I'm more interested in continuing the titration experiment and adding more base, and that will let us start working towards the next titratable group, which is the amino. And notice how, since an amine is more basic and thus more thermodynamically inclined to remain protonated, it requires even more base to deprotonate. And like the carboxyl, the amine will act as a good basic buffer near its second pKa, which dials in, as expected, at around 9 pH units. At the final endpoint, after 2 equivalents of hydroxide was titrated, we are now left with a fully protonated form of glycine, where we lose the positive charge on the amine and retain the negative charge on the carboxyl, giving us a glycine with a net -1 charge. There's a lot that goes on with these titration curves, so I encourage you to pause, rewind, and think as necessary to become confident with now connecting the protonation states of our amino acids to the titration curve. Time to crank things up. I'm showing you another titration plot, and I haven't given you printouts of the amino acids. You can consult the internet, but generally describe how would you identify the amino acid being titrated here in this experiment. All right? Just going off the curve alone. The way I like to approach these problems is looking at their pKa's. Recall that the pKa's of the N and C termini to all amino acids will be respectively 9 and 2. Follow along, and we can identify that pK1 and pK2 correspond to the buffering zones of the amine, the N terminus, and the C terminus, the carboxyl group. These will be present in the titration plots of every amino acid, so they're not going to help us identify the unknown here, but it will help us highlight pK3. This is the key to solving the mystery. If we move across and find its pKa value, it looks to be somewhere between 10 and 11. And this is why it's important to have your amino acid sidechain pKa's memorized, because that is all you need to identify it from here on out. But we can reason through a little bit further and state that since the third pKa requires enormous amounts of base to deprotonate it, we can assume that it is basic, so that narrows the pool down to arginine, histidine, and lysine. And I happen to know that the pKa of lysine is 10.5, so this would be lysine. It's good to know how to do this skill. Now I want you to pause and draw out lysine and predict what you think the charge and protonation state would be of all 3 protonatable groups at the beginning of this experiment, where the pH is really low and we haven't added much, if any, sodium hydroxide yet. I'll pull up this diagram over here that we can work alongside with, and I will show you that at very, very acidic pHs, denoted by this red star, the carboxyl, the C terminus, is protonated and neutral. The N terminus is protonated and positively charged. The epsilon amine is also protonated and positively charged, making the net charge on lysine 2+. Ask yourself, of these three entities, which is the most acidic? The most acidic group will be the most easily deprotonated by the hydroxide we're adding in. And I'm noticing that the carboxylic acid is going to be quite acidic. So it comes off pretty easily at pH 2, and we transition from COOH to COO-. We are 50-50 red star and beige star at the pK1 points, and we become 100% lyse positive over here at this equivalence point. Moving along the curve, the second most acidic group after the carboxyl will be between the N terminus and the amine on the side chain. This has a pKa of 9, this has a pKa of 10.5. So that means this is the next one to lose its proton. And when we deprotonate an amine, it goes to neutral. So we've lost a positive charge. The negative charge in the carboxyl and the side chain amine cancel out, and lyse neutral will become apparent at the isoelectric points. It will be 50-50 beige and this periwinkle star at the pK2 points. And I'll write that again over here. At the isoelectric point, it's 100% lyse neutral. The only other titratable group we haven't touched yet is the one on the side chain, the most basic one, the least acidic one. That's why we need so many equivalents of hydroxide added. 2.5 equivalents of hydroxide is quite a lot. That is where we reach the 50-50 point between light blue and regular blue. And only when we go beyond that do we completely strip the protons from lysine and generate lyse negative. If you can master this, you will be in good shape in correlating protonation state with the pH of the solution the amino acid is in. I think we can agree that the protonation state of an amino acid is fluid. The charge on any of these amino acids varies greatly across pH. There are some constants worth considering, though. Let's recall that the alpha carbon of an amino acid is tetrahedral. That means it's a potential stereocenter. This leads us to maybe ask, are amino acids enantiopure, or do they exist as racemic mixtures? There are two ways we can investigate this question. First, let's throw back to the Kohn-Ingold rules from Orgo 1. If you have, let's say, the structure of a compound, we learned to assign priority to different functional groups and moieties depending on if priority proceeds clockwise or counterclockwise about the axis between the alpha carbon and the hydrogen group that bonds. And depending on if it goes clockwise or counterclockwise, we can say if an amino acid is R or S. I have a amino acid shown over here. It is going to be alanine because there's a methyl group. And is this R or S? Well, we assign greatest priority to the amine, second most priority to the carboxyl group, and the methyl group will be the lowest priority. So this proceeds in a counterclockwise fashion. That means we have an S amino acid. As a matter of fact, all amino acids are S. This can get tricky because there are some amino acids, like isoleucine, that have a second stereocenter in their R chain. And so the Kahn-Ingold system is actually not a terrific tool for describing the chirality of amino acids. Instead, you can get a better feel for a molecule's chirality through a technique called polarimetry. Here in polarimetry, we will pass a beam of plane polarized light through a cuvette containing an allegedly chiral compound, like an amino acid. And as the plane polarized light comes through, if it's chiral, the beam will optically rotate clockwise or counterclockwise, to the left or to the right. And every amino acid rotates to the left. Don't be fooled. Just because a compound optically rotates to the left doesn't mean it is the S enantiomer. It just so happens that every amino acid on the planet is the L enantiomer, and that is very important. I should also quickly mention that glycine is an exception because the R chain is also hydrogen, which means we have an internal plane of symmetry and we lose all handedness. So glycine is achiral, but everything else is S and L, enantio pure. Even the amino acids found in space? This is actually pretty cool. In 1969, a meteor exploded over Australia. It's called the Murchison meteorite. And scientists collected and analyzed these samples and were able to extract out a slew of complex organic entities, including several amino acids. What's more surprising is that these amino acids were found to have an L enantiomeric excess between 2 and 9%. This is significant because it suggests that the interstellar medium has the conditions needed to induce or prompt homochirality. Life is homochiral. All proteins contain amino acids that are in the L configuration, with very, very few exceptions. But some scientists were understandably skeptical of this finding because we live in a homochiral world and only a few nanomoles of amino acids were extracted from the meteor fragments. Can we confidently say this was a spontaneously generated excess? Or was this contamination from planet Earth that skewed the results? Well, a few years later, this was 1969 after all, we repeated the experiments off the same meteor fragments with newer and better and more sensitive technology. And in doing so, we discovered some extraterrestrial amino acids, pseudoleucine, isovaline, and norvaline. These have not been discovered anywhere on Earth, at least naturally to my knowledge. The only place they exist is in outer space or the Murchison meteorites. And all of these also existed in a 2 to 9% L enantiomeric excess. This means that somewhere out there deep in space, the universe can break chiral symmetry abiotically, which is quite impressive. If we were all to go to the lab right now and try to make alanine in a flask, that's quite easy to do. The problem is, when scientists make amino acids, we get a racemic mixture, a 50-50 distribution of one enantiomer to the other. And biology does not like that. So the fact that this can happen on a meteorite hurling through space is huge. We covered a lot of ground in this introduction to amino acids. The question still persists, why has nature selected them to be the monomers of proteins? If we were to condense the information we've covered into 3 points, we could perhaps argue that an amino acid's 3 critical properties are: 1, their heterobifunctional backbone, which is to say their presence of N and C termini. Second, their diversity of sidechains, R groups. And third, the fact that they are homochiral. They are 100% in the L configuration. So my last question is why? How do these properties of amino acids make them so well suited as protein monomers? This will be a discussion that we will have in class, but I would like you now to pause the video and try to come up with answers, prompts, and counter arguments that you can bring to the table in class. I'd encourage you to do a little bit of Googling and researching. Don't feel like you need to bring in a report with a bibliography, but maybe have some examples in your back pocket, which may make the discussion more stimulating. As a bonus, in this slide and the next slide, I'll provide you some reference materials that may aid in your study. This is the world's most complicated Venn diagram that shows the shared properties among all of the proteinogenic amino acids, prepared by Lauren Williams of Georgia Tech. And in this slide, I have the skeletal structures for the proteinogenic amino acids conveniently compiled into one place, along with their names and their single letter codes, their sidechain pKa's where relevant, and it's all color coded. So this, I think, is a great place to begin committing this all to memory. I also highly recommend flashcards. They exist as free apps and browser games that you can access on your smartphone. You can study on the go and quiz yourself on the amino acids between classes. That's what I did when I was in college. I will also say, I'll caution you that recognizing an amino acid is way easier than drawing one from memory. At this point, perhaps you could say, oh, this amino acid is lysine. But if I were to ask you on a test, draw me lysine from scratch, how many of these methylene groups are there between the alpha carbon and the epsilon amine? That may not be very obvious if you haven't fully committed the structure to memory. So word to the wise, before our exam, by next week, we will be name dropping and referencing and drawing amino acids willy-nilly. And you may have a hard time if you haven't completely absorbed them by then. So I would put a lot of efforts before next lecture and over the weekends for sure to getting these down pat. So good luck, study hard, and I'll see you next time.